So thank you, Karl-Heinz, for the introduction. And I want to shift gears from neural networks to all the way down to ion channels and dendrites in single neurons. So I'm from Geneva Research Campus, and I work in Nelson Brusten's lab as a postdoc associate. And our lab studies the ion channels, dendrites, and the hippocampal function in the brain. So today's talk is not about a particular project, but it's a relatively higher level introduction into these two components of computation. So in the brain, it contains a vast number of different cell types with distinct morphologies and perform distinct physiological functions. So the pyramidal cells within these different cell types are particularly important because their prevalence in the higher function brain regions, such as the neural cortex and the hippocampus. And so here is a picture of the red hippocampus, and the immunolabeling of green is CA1 pyramidal cells. And the red labeling is the parvalbumin expressing inhibitory cells. So I just wanted to give you an impression on how complex the network is. And to understand this complex connectivity between pyramidal cells and inhibitory cells, we have to understand how a single neuron computes. So to start to understand a single neuron, let's go back to the first principles and start to think about what really neuron is. So at the most fundamental level, neurons are really lipid bilayers in a salt solution. So these are insulators with ions flow inside and outside the cell. And through evolution, these ions are being separated by membrane proteins called transporters and ion channels. And these transporters can separate different charges, and ion channels can pass different ions through the membrane. So the end result of the evolution is that the neuron typically has a resting potential around -70, and there's a negative charge near the inside of the membrane, and there's a positive charges at the outside. And different ion channels are being scattered around the membrane to modulate locally the electrical activity. So the input-output function of a neuron can be understood like this. So on the dendrites of a neuron, it receives neurotransmitter release from the presynaptic neuron. And this neurotransmitter opens these ion channels and allow current to go into the dendrites. And this current is being transformed through a function. And that function can induce eventually an action potential in the axons of the neuron. Action potentials can travel down to the axonal terminal, and it can raise calcium concentration in the axonal terminal and induce neurotransmitter release onto the downstream neurons. So this process is really like an alternation between analog signal and a digital signal. So what is exactly the input processing function of all those currents coming into the cell? So a simple model is the McCulloch-Pitts neuron. So the assumption is that the different inputs are summed linearly and then pass through a nonlinear activation function to give you an all-none output. But this simplified model does not capture some of the key features of real neurons. So the two key components shaping the input processing function in biological neurons are ion channels and dendrites. So these will be the focus of my talk today. So let's first focus on the ion channels. So before ion channel, let me introduce, so I'm plotted here is a representation of the cell membrane. It's a lipid bilayer. It's a good insulator. And the cell membrane has a relatively thickness of around 4 nanometer. And through evolution, different ion channels, proteins, are being inserted in the membrane to do different things. And here I'm just plotting a variety of ion channels typically seen in the neuronal membrane. So you can see this ion channels typically has the diameter around 10 nanometer and looks pretty complex. So let me orient you to the part where ion channel is going through the membrane, which is called the transmembrane domain. So these transmembrane domains inside the membrane contains a pore which has selective filters for different ions of different diameters. And that's responsible for current flow. And all these extracellular and intracellular big chunks of protein modulators are evolved through different ways to either gate the current or modulate the current in different ways. So ion channels are really the basic computational elements in neurons. Because they can pass ions, pass currents through the membrane and modulate locally the potential across the membrane. So today I want to focus on the three major players during input transformation in neuronal processing. So the three proteins are voltage-gated sodium channels, and voltage-gated calcium channel, and the NMDA receptor. So I picked these three channels because they share a common feature. So these three channels are all voltage dependent. So the reason they are being voltage dependent is because their transmembrane domain here, either for the voltage-gated sodium and calcium channels, it contains an array of positive charged amino acid residues like arginine. Or for the NMDA receptor case, there is a positive charged magnesium ion stuck in the pore to block the current flow. So to gate the channel, to make it open, during depolarization, the membrane potential inside, there are positive ions going into the membrane and depolarize the cell. Once the cell is depolarized, it induces conformation change of this positive charged amino acid residues, and then cause a conformation change to gate the channel to open. For the NMDA receptor case, the magnesium block is being repelled out of the channel pore. So now sodium and potassium can go into the cell and do electrical signaling. So these voltage dependence is critical for all the nonlinearities in neuronal function. So as an electrophysiologist, we typically record these channels by voltage clamp recording. So basically, we clamp the channels at different voltages and record how big a current it gets flow through the membrane. And if you pay attention on the right, so that's IV, the current voltage relationship of this current. So you can see that the current amplitude at relatively higher voltages, the current and voltage relationship is around linear. And that's because the channels open all the time at positive voltages, and the linear relationship is basically a reflection of the Ohm's law. However, once the voltage starts to decrease at negative voltage where the cell is resting at, the current starts to shut off. And then at really hyperpolarized voltages, there's no current. And the same is true with voltage-gated calcium channels and NMDA receptors. So you see this J-shaped IV curve a lot in electrophysiology. So the basic idea is really Ohm's law, a linear resistor current convolved with a Boltzmann activation function. So let me just go through these three different channels one by one and discuss their functions and impact on dendrites computation. So first, I want to say something about the regular sodium spike, the action potential. So the voltage-gated sodium channels are responsible for the generation of action potentials, along with the voltage-gated potassium channels. So these channels are concentrated near the cell body and on the axons of the cell. So if you think about the J-shaped IV curve of sodium channels, so once the memory is depolarized, it starts to let sodium into the cell. And that in turn increases the voltage of the cell and in turn opens more channels. So this is a positive feedback loop. And you can imagine that can cause a regenerative event like the action potential. And indeed, if you inject current in the cell body, and once the membrane potential reach a certain threshold, there's irreversible all-or-none action potentials happening at the cell body and axons. So it has also been observed that the voltage-gated sodium channels are also expressed at high levels on the dendrites. And that's really counterintuitive because dendrites were viewed to be passive integration zones for neurons for a long time. And these voltage-gated channels on the dendrites really made people think, okay, are they processing information in a nonlinear manner as well? So really not until early 90s, we start to be able to address this type of questions because of the development of dendritic recording technique. So we can put electrodes on the dendrite itself instead of the soma and record the electrical activity on the dendrite and the soma simultaneously. And this recording done by 98 is showing that, so if you pay attention to the thicker black traces and the thinner traces, are the voltage recording either on the dendrite or on the soma. So after a brief stimulation to cause neurotransmitter release, you can see that the dendrite has an electrical change that's higher than and propagates to the soma. And at one point here, the dendrites start to spike. And this is all or none. And before the soma even spikes. So this is one of the first evidence showing that the dendrite can spike on its own without even a back propagating action potential, because the last figure showing that after the dendrite spikes, the cell starts to spike. So what could be the computation this local dendritic spike is doing? So one simple idea is that different dendritic trees receive different inputs. So you can receive distal inputs at the upper part of the cell and receive a proximal input at the proximal part of the cell. So these two inputs can be from two different brain regions or two different information flow into the cell. So one computation it can do is that it's a coincidence detection. So it can only allow the cell to fire once the two inputs are coactive. So here I'm going to show you a simulation on the distal input inducing a local dendritic spike. So the right trace is the local membrane potential of the distal dendrite. So you can see that some input causes a local spike, but this spike does not reliably propagate to the rest of the cells. And also, if we now do a simulation of the proximal input, we just release some neurotransmitter on the dendrites, and we can see that this is a relatively weak input. So it just depolarized the part of the dendritic tree a little bit. However, if the two things are happening simultaneously, you can see that the distal starts to fire and the proximal fires and immediately cause the action potential in the cell body. So this can be so let's look at it again. Distal and proximal fires and the cell fires. So these could be a mechanism of using another input to gate whether you want to propagate the signal out of the cell from the distal input. So the second dendritic spike people observed is so-called the calcium spike. And calcium spike is caused by the voltage-gated calcium channels. And similarly to sodium spikes, it can also do coincidence detection. So here is an early recording from Matthew Lockham in layer 5 pyramidal cells. So he put 3 recording electrodes either at the distal part of the dendrite or the proximal part, and also another one at the cell body. And it has been shown that voltage-gated calcium channels are heavily expressed at the apical trunk of the dendrite. So here's a complex figure. I'll go through it slowly. So here is the membrane potential of the red electrode. So here is the I stim is how much current you're injecting into the dendrite. And if you inject a little bit of current, you cause some depolarization in the distal dendrite. And here is referring to the black electrode in the cell body. If you inject a square pulse of current injection, the cell body can generate action potential, which is the black trace. And you can ignore the backpropagation part of the action potential. So now, if you pair these two current injections together, so you inject with the red electrode a small depolarization at the distal dendrite and a square pulse injection in the soma, and then something different happens. So instead of only firing one action potential, the cell fires a burst of action potentials. And that's because if you take a look at the red trace is a very broad, so-called plateau potential. And this is another type of local dendritic spike. And these two coincidence events can be detected through the dendritic spike and cause a different pattern of firing of the cell. So it seems that the cell is capable of doing either a single spike or a burst of spikes, depending on the stimulus pattern. And I think this is a very interesting way for the cell to multiplex. So imagine that. So sometimes the cell fires a single spike, and sometimes the cell fires a burst of spikes. And they might encode different information. But how does the downstream target know which information you are encoding? And a really elegant solution is actually at the synapses. So typically synapses are divided into two different types with two different dynamics of short-term plasticity. So one is called the depressing synapses. So with multiple action potential invasion, the neurotransmitter release probability actually drops dramatically. So the first pulse, the neurotransmitter release a lot, and then it decreases. The second type is the facilitating synapses. So it barely releases anything if there's a single spike, but it releases a lot of neurotransmitter if there's a burst. So you can imagine these two types of synapses can convey these two informations to different brain targets for different information processing. And at last, I want to introduce something about NMDA receptor. So NMDA receptors are very, very unique in a way that it's both a ligand-gated ion channel, and also it's a voltage-gated ion channel. And what I mean is that, so in order for the channel to open its pore, it needs to bind a neurotransmitter. And in order for the ion to pass through the pore, the magnesium needs to be removed, and you need depolarization. And itself is a coincidence detector. So it has been shown that NMDA receptors can also generate regenerative NMDA spikes in local dendrites. So here's an early experiment done by Jackie Schiller. You just pay attention to the middle figure here, that if you record from the cell body and then stimulate the axons passing through the basal dendrite, with increasing stimulus intensity, there are more and more neurotransmitters being released. And you can see that at the beginning, the amplitude rises slowly. And at one point, the amplitude sharply goes into an all-or-none regime. And there's this large, long-lasting NMDA spikes happening. So just the summary that all this information is pointing to this general picture of how a pyramidal neuron works. So voltage-gated sodium channels can generate sodium spikes throughout the cell body, which is indicated in the cell body in the dendritic trees, which is indicated in blue. And voltage-gated calcium channels are shown in red, and typically they only generate near the bifurcation of the apical dendrite. So this is important functionally. And the third one is NMDA spike. So this spike happens all over the thin dendrites. And the thin dendrites consist of the majority of dendrites, like 80% of dendrites of a pyramidal cell. So the emergent picture is, so we can view a single pyramidal cell, each individual branches, as an individual active computational element. It can sum inputs and go through a nonlinearity. And also this pyramidal cell has multiple integration zones. So each individual NMDA spike, for example, can go to the bifurcation and cause a calcium spike. And this calcium spike and a somatic spike can multiplex or generate a burst or single spike, depends on the configuration. So really, as we can look at the single neuron almost like a 3-layer neural network instead of a single artificial neuron. So at last, my conclusion is, so ion channels are very complex computational machines. So it's only around 10 nanometer. It contains many, many domains. It uses all the forces nature give it. Salt bridges, hydrogen bonds, ions. And its behavior is very complex. And we typically describe this behavior by a Markov model that looks like this. It contains many states. And the second is point neuron model does not fully capture neuronal function. And dendritic spikes can really increase the computational power of a single neuron. So there's one point I want to share, I think is potentially important and need more attention on NMDA spikes. So NMDA spikes is really unique due to the double-gated mechanism, ligand gating and depolarization. And there's this very strange state. It's called agonist bound but blocked state. So you can have neurotransmitter bound to the channel. It's open, but there's a magnesium ion in the pore blocking the channel. And this is, I think it's profound because NMDA receptor has a very long kinetics. So after a 1 millisecond glutamate release, it opens for hundreds of milliseconds after the glutamate is completely gone. So after the glutamate is bound to the receptor, it's primed, but it's magnesium blocked. So with magnesium, you record no current. And this primed state can encode very important things in the dendritic branch. So at different times, glutamate can prime different branches with glutamate, but those branches, and enable those branches to be potentially active. And these branches, so NMDA spikes and glutamate release can really do online reconfiguration of circuits with minimal energy cost because ligand binding, it only requires ligand binding and with minimal current flow. So with that, I want to thank my advisor and my lab mates, and I want to thank you for your attention, and I won't take any questions.
Thank you, Weinan. Are there questions? Yes, please.
So I have a question on, so what you've described here is the sort of, different parts of the dendrite process information differently, and they can interact in very sophisticated ways. And so, we had a short paper on how different layers, inputs can change computation. Do you know of, but that was entirely theoretical. Do you know of experimental evidence where, like a clear computational story where these pyramidal neurons are actually using different sources of information to do a computation that most people here would not appreciate that one neuron can do? Like specific experimental evidence to that effect.
Yeah, so I think the closest thing I can imagine is the C1 pyramidal cell. So the distal part receives a very distinct input from the entorhinal cortex and from different parts of the brain. And the proximal part receives inputs from the CA3, where's the Hopfield net. And I think this is a critical computation that the hippocampus is doing the pattern separation and assigning like a distinct attractor to this CA3 axons, and in the proximal part of projects the proximal part of the CA1. And then that can be paired with information-rich a distal input to the distal part of the dendritic tree, and then combine these two. So you can have pattern separation, distinct memories, and also you can recall, you can add information to that code as the CA1 output of the hippocampus to the rest of the brain. And there might be other studies I'm not aware of.
Can I ask you a question? You said that the pyramidal neuron is kind of a 3-layer network of point neurons. So would you say that you could computationally replace a complex neuron just by a 3-layer network of simple point neurons?
Yes, people ask me the first question after I say that.
That's the obvious question that everybody asks.
I don't think so. So because
give us the reasons.
I had an argument with one of my colleagues. He said, okay, these neurons are universal Turing machines, and you can wire anything. Why do you need dendrites? I think 2 arguments. So the first argument is, so if I give you a pyramidal neuron with that connectivity, with different ion channels on there, and I'll give you the task of, okay, draw an equivalent circuit of different layers of point neurons. I think that could be computationally intractable because these different ion channels interact with nonlinear ways, and the branches is doing very complex stuff with time dependence as well. And secondly, even if you can solve the problem of writing that diagram, it probably requires tons of hundreds more of cells. So it's energetically not feasible. So that's my argument to that.
Okay, thank you. Certainly a point maybe for the afternoon discussion.
Okay.
So thank you, Weinan, again for your